It's a great pleasure to introduce Dr. Tiffany Ho. Dr. Ho is an assistant professor in the Department of Psychiatry and Behavioral Sciences at UCSF and is the director of the Cognition, Affect, and Neurodevelopment in Youth CANDY Lab. Dr. Ho received her bachelor's degree in cognitive science at UC Berkeley before earning her PhD in psychology and cognitive neuroscience at UC San Diego. Prior to joining the faculty at UCSF, Dr. Ho was a postdoctoral researcher and instructor at Stanford University. Dr. Ho's work in the CANDY Lab focuses on using multimodal neuroimaging cognitive and behavioral assessments to understand how stress affects neurodevelopment in ways that place adolescents at risk for developing depression and other related conditions. Please join me in a warm welcome for Dr. Tiffany Ho. Take it away. I'll mute myself now so you can start. Thanks for being here.
Oh, thank you so much for having me. Okay, so let me share my screen. All right, well, thank you all for having me. I'm really delighted to be talking about my recent work focused on stress and neurodevelopment adolescence, and specifically the roles of inflammation and glutamate. Let me swap this. There, that should look better now. So to begin, I have no biomedical conflicts of interest to disclose. My research has been funded by the Klingenstein Third Generation Foundation, the National Institute of Mental Health, and the Ray and Dagmar Dolby Family Fund. I'm hoping by the end of this talk, you'll gain more knowledge about the general neuroscience of adolescent stress and depression, as well as the roles of inflammation and glutamate in adolescent brain health, and potential treatment strategies or adjunctive strategies for improving adolescent mental health in the context of stress and depression. So as an overview, I will begin by talking about the public health relevance of adolescent depression. I will then launch into a more detailed discussion of an ongoing project that I am directing, the Teen Inflammation Glutamate Emotion Research Study, also known as TIGER. TIGER is a KO1 award funded by the National Institute of Mental Health, and it's focused on understanding the roles of inflammation and glutamate in adolescent stress and depression. I'll then briefly talk about some implications of the work and future directions before we go into a wider discussion. So adolescent depression, why should we care? As you all know, depression is unfortunately relatively common, severely debilitating, and it represents a huge societal burden. It is currently the leading cause of disability in individuals in the United States between the ages of 15 and 44. And here is actually a graph of relatively recent data demonstrating depicting the projected cumulative incidence of depression in individuals between the ages of 12 and 17, plotted by a function of biological sex. And so by age 18, one can expect about 1 in 4 adolescents to experience a clinically significant episode of depression, with rates unfortunately being twice as high in females as compared to males. Of course, the data I just showed was prior to the COVID-19 pandemic. So a recent meta-analysis published earlier this year, unfortunately, showed that rates of depression and anxiety across the globe have doubled compared to pre-pandemic times. So why is adolescence a vulnerable period for depression? Adolescence itself is a dynamic time characterized by tremendous neurodevelopmental and neuroendocrine changes that are coupled with an increase in variety of stressors and shifts in social identity, moving more towards independence. And I think, as a lot of us know and can recall, stress is a potent trigger for the onset of depression during this age period. And recent studies have actually documented that interpersonal life events or interpersonal stressors specifically have been found to be statistically unique predictors of the subsequent onset of depression in a variety of study cohorts. And in an ongoing study from my lab, we're also finding that social stress severity, so these are experiences that are typified by interpersonal conflict. So that includes peer victimization, social rejection, romantic breakups, things of that nature. Greater severity of this type of social stress, but not non-social stressors, related to higher levels of depression at baseline as well as over the course of 9 months, and was uniquely related to lower gray matter volumes in a variety of brain structures that are related to emotional processing and regulation. So that includes the anterior cingulate cortex, shown here in blue, the ventral medial prefrontal cortex, shown here in purple, as well as limbic structures, including the amygdala and the nucleus accumbens, shown here in green and red, respectively. And what we found was that reductions in gray matter in these frontal cingulate limbic structures not only predicted depression severity over time, over 9 months, but it also statistically mediated these associations between social stress severity and depression. So essentially, what we're finding is that these adolescent-specific stressors that are characterized by social threat, these are occurring against a backdrop of neurodevelopmental and neuroendocrine changes that could render specific neural circuits that are underlying emotional processing and leading teens to either develop depression or to maintain a depressive state. Despite adolescence being a high-risk period for certain mental health conditions, I do want to emphasize that this is also a time of potential resilience. And as leaders in adolescent brain health have recently written in a perspective piece in Nature a few years ago, adolescence is a period of rapid growth, learning, adaptation, and formational neurobiological development. It is a time when lives can pivot rapidly in both negative as well as positive directions. And so it really speaks to the importance of investing during this time and cultivating positive environments and healthy and adaptive social interactions so that the greatest gains really can be made in terms of longer term health, education, and socioeconomic outcomes. So as a quick interim summary, depression is unfortunately alarmingly prevalent in adolescence. And it's also a condition that tends to be chronic and recurrent. So that speaks to the need to understanding how adolescence or how depression emerges during this time period. Adolescence is also characterized by significant physical and neurodevelopmental changes coupled with shifts in social identity. And as a result, stressors occurring during this time may have a disproportionate impact on the development of neural circuits that are relevant to emotion regulation and that underlie the development and maintenance of depression. That said, I think the field currently is lacking a more mechanistic understanding of how stress is affecting the developing brain in ways that may place teens specifically at risk for depression. And so it was with this motivation in mind that I started the TIGER study. So TIGER again stands for Teen Inflammation Glutamate Emotion Research. And the main tenets of TIGER is to test whether or not inflammation specifically is a stress-related driver of depression through its effects on the developing brain, and whether glutamate is one key neurotransmitter by which this process occurs. And so here again is a schematic proposing the scientific model for TIGER. So the purpose of this study is that we will be looking at levels of inflammation, particularly pro-inflammatory cytokines such as interleukin-6 and tumor necrosis factor alpha in the periphery, and seeing whether or not heightened levels of inflammation are related to glutamate in frontal cingulate limbic circuits, and whether or not the confluence of inflammation glutamate are contributing to depression symptoms in adolescence. This model of inflammation and glutamate is really one that's informed by basic science, work on the fundamental biology of stress. And several studies have shown that Psychosocial stress activates the immune system in the same way that a pathogen-initiated response would. And that then leads to increases in inflammation that in turn can impact the brain through a variety of complex mechanisms. Andy Miller at Emory published a lovely review paper in Biological Psychiatry in 2009 that outlines a lot of these complex pathways in more detail. But I want to turn your attention to two primary ones for the purposes of this talk. So the first is the kynurenine pathway. The kynurenine pathway is a metabolic pathway. And so when an organism is experiencing psychosocial stress, the immune system mounts a heightened inflammatory response. And that includes the secretion and production of pro-inflammatory cytokines, such as interleukin-6, tumor necrosis factor alpha. These cytokines can cross the blood-brain barrier, where they are interacting then with microglia, depicted here by this blue blob. And microglia represent the first line of immune defense in the central nervous system. When microglia are activated, that upregulates an enzyme known as indolamine, which is a critical player in the kynurenine pathway. Indolamine breaks down tryptophan, which is a precursor to serotonin, into an acid known as quinolinic acid. And quinolinic acid is a potent NMDA receptor agonist. So you see quinolinic acid here binding to NMDA receptors, and that leads to a release of glutamate into the synapse. Over time, so glutamate obviously is an important excitatory neurotransmitter, but over time, too much glutamate can lead to what is known as glutamatergic excitotoxicity and neuronal cell death. So you can kind of imagine it's like a circuit that short circuits. And with that, you will also find a reduction in brain-derived neurotropic factors. And I posit that chronic stress can then lead to excessive glutamate and an impact on gray matter that can be observed at a more macro level through reductions in surface area, cortical thickness, and gray matter volumes. And of course, associated alterations in functional connectivity in relevant regions. At the same time, another relevant pathway that I term inflammatory demyelination can also lead to excess glutamate in the central nervous system. So when peripheral cytokines interact with the central nervous system and microglia, that also initiates a cascade of inflammatory responses in the central nervous system. So that includes increases in cytokines, chemokines, inflammatory mediators, and oxidative stress. When that occurs, these markers are also affecting the integrity of astrocytes, depicted here by this green star-like structure, and other glial elements, including oligodendrocytes, which comprise the myelin sheath. And one major function of astrocytes, in addition to providing neurotropic support to oligodendrocytes, is to take up excess glutamate in the synapse and to convert glutamate into glutamine, which is a precursor for glutamate and GABA. And so you can already see here, there are two pathways by which inflammation can lead to excess glutamate through the kynurenine pathway, and as well as through the inadequate reuptake of glutamate, because the integrity of these astrocytes have been compromised by these inflammatory mediators. And I posit also that over time, we should be able to see reductions in white matter, and of course in associated functional connections in relevant structures as a result of stress-related inflammation. So in terms of applying this inflammation glutamate model from a stress perspective to depression, non-human animal studies have shown that increasing inflammatory cytokines through experimental manipulations leads to behavioral phenotypes that resemble aspects of depression. So that includes anhedonia, social withdrawal, and fatigue. In terms of human studies, Ibrahim Haroon and Andy Miller at Emory have shown that injecting interferon alpha in non-depressed adults increases glutamate in a region of the anterior cingulate cortex, specifically the dorsal anterior cingulate cortex. In another study by George Slavich and colleagues at UCLA, healthy adults underwent a psychosocial stress test that included social rejection. And they found that this stress test increased salivary levels of pro-inflammatory proteins, as well as the soluble receptor for tumor necrosis factor alpha. And higher levels of this immune or stress-related response was strongly and significantly correlated with activation to social rejection in the dorsal anterior cingulate cortex. So what these studies here are showing are that inflammation does seem to be impacting glutamate in the brain, and specifically in an area of the anterior cingulate cortex that may be sensitive to social stressors, and that may be also a target of these processes. And I find this work interesting because it dovetails with my previous neuroimaging studies where I sought to examine the neural correlates of adolescent depression. And in those studies, I found that a lot of the evidence converged on this frontal cingulate limbic circuit. And so that includes the anterior cingulate cortex region, shown here in blue, the ventral medial prefrontal cortex, shown here in purple, as well as limbic structures such as the amygdala and hippocampus, shown here in green and teal. And together, this circuit is really involved with not only emotion processing, stress regulation, but in general, the processing of salient information, both negative and positive, and integrating past experiences to produce behavioral outputs. The Tiger study is focused on the anterior cingulate cortex, in particular the dorsal anterior cingulate cortex, just given the previous work that I noted. And I want to highlight that the anterior cingulate cortex enjoys many structural and functional connections with these other frontal and limbic regions, and as a result has been a prime area of focus for many studies on depression. Very quickly, I do want to highlight one such study that I conducted with Dr. Tony Young here in the department, where we found that functional connectivity of the dorsal anterior cingulate cortex was aberrantly inflexible in depressed teens as compared to healthy controls. And what I mean by aberrantly flexible is that connectivity in this region did not change in depressed teens as a function of environmental demand. So as you can see here in this bar plot at the bottom, healthy controls showed great shifts in connectivity of the dorsal anterior cingulate cortex, depending on if the teens were at rest versus engaged in a cognitively demanding task. So there's a big difference here. However, surprisingly, in some ways, the depressed teens did not show any such responsivity or engagement with the environment as a function of task demand. And so we posited that this underlies non-adaptive forms of cognitive processing and may even reflect atypical neurodevelopment, since this is not what would be expected given more typical neurodevelopmental trajectories across this age group. And indeed, we actually found that greater aberrant connectivity of the dorsal anterior cingulate cortex in depressed teens was associated with an earlier age of depression onset. In other recent work that I've done, I've also shown that frontal cingulate limbic network connectivity, as defined using a more data-driven approach, was significantly reduced in depressed teens as compared to healthy controls. And similarly, the integrity of white matter tracts underlying or connecting bilateral frontal cingulate regions was also reduced in depressed teens versus healthy controls. So again, I want to highlight that even though Tiger is focused on the dorsal anterior cingulate cortex, there is really a constellation of regions at play here. And the focus of the dorsal anterior cingulate cortex is due to past work, as well as the fact that this is a region that enjoys multiple connections with networks that are broadly implicated and relevant for depression. So to bring this all together, here's a quick schematic describing the theoretical model and the specific hypotheses that we're hoping to test with Tiger. First, we want to look at whether or not higher levels of peripheral inflammation are related to higher levels of glutamate, specifically in the dorsal anterior cingulate cortex. And then we want to then see whether or not these markers are also associated with altered frontal cingulate limbic connectivity. And Tiger is also a longitudinal study, so we will be collecting information on depression over time. And I also wanted to be able to test whether or not inflammation in glutamate may be related to depression recurrence. As an exploratory aim, I'm also interested in testing whether or not antioxidants in the anterior cingulate cortex may be moderating any of these hypothesized associations. And we are using a very unique spectroscopy scan that will allow us to measure and estimate antioxidative capacity in the brain, and I'll talk about that more later. So the scientific rationale and the design and protocol of the Tiger study has been published last year in Frontiers in Human Neuroscience. So for those who are more interested in all of the details, I urge you to check out that paper. But briefly, I will summarize the design here. We're hoping to recruit 75 clinically depressed teens between the ages of 13 and 18 who meet diagnostic criteria for major depressive disorder and who are post-puberty in status. Exclusion criteria includes meeting for lifetime criteria or endorsement for psychosis, mania, or substance dependence. And as I mentioned earlier, Tiger is a longitudinal study. It will be 18 months long, but it is naturalistic observation. And so over the 18 months, we will invite participants to come in at 3 time points, time 1, time 2, time 3. Each time point will consist of 2 visits, approximately 1 to 2 weeks apart, where participants will undergo clinical interviews, self-report questionnaires, cognitive tests, a multimodal MRI scan, including a spectroscopy scan to image glutamate. And they will also provide blood samples from which we will assay peripheral cytokines. Every other month in between time points, we will also ask participants about their depression and anxiety symptoms, as well as any applicable treatment changes. And the goal here is to not only ensure engagement and contact with our participants, but also to be able to model trajectories of depression symptoms, as well as to identify potential periods of depression remission, as well as recurrence. So here is a summary of all of the different measures that we're getting across several domains: clinical psychological constructs, multimodal imaging, cognitive tasks, and inflammatory cytokines. I think in the interest of time, I will move forward, but if you're interested in the details of specific measurements, please read our protocol paper. To measure inflammatory cytokines, we are using contact-initiated lancets to prick the fingers of our participants in order to collect blood drops, as opposed to more traditional venipuncture methods. These blood samples are collected on specialized filter paper, which is then dried overnight using procedures developed by my consultant Tom McDade at Northwestern University. So an obvious advantage to dried blood spot technology is the ease of data collection or blood collection and storage compared to traditional venipuncture. In collaboration with Holden Macker and Yale Rosenberg Hassoun at the Human Immune Monitoring Center at Stanford, we've been testing the extent to which multiplex assays or panels on dried blood spot samples allow us to estimate concentrations or levels of various inflammatory proteins and whether or not they are reliable and correlate strongly with those obtained from plasma. And we were finding that interleukin-6 and tumor necrosis factor alpha do show such strong correlations. And based on meta-analyses in the field, these are actually two markers of inflammation that have been strongly related to depression, both in adolescents and adults. So even though the panels that we are running allow us to theoretically look at potentially up to 60 different analytes or proteins, for the sake of the data, as well as theoretically what we've been seeing in the literature, our analyses are going to be focused just on interleukin-6 and tumor necrosis factor alpha. With respect to our neuroimaging portion of the study, we are collecting a series of structural and functional scans that will characterize gray matter, as well as white matter and intrinsic or resting state functional connectivity. And of course, we are also acquiring a proton magnetic resonance spectroscopy scan that will be focused specifically on the dorsal anterior cingulate cortex. Unlike the other scans, which cover the entire brain, and for the purposes of boosting signal-to-noise ratios, we're using a sequence that's focused on a specific region or voxel. So the proton magnetic resonance spectroscopy scan currently is the only way we can non-invasively image neurotransmitters such as glutamate and related metabolites. And this sequence that we're using was developed by Dan Spielman in the Department of Radiology at Stanford. And it will allow us to look at not only glutamate, which is our primary measure of interest here, but also glutathione and ascorbate, or also known as vitamin C, which are antioxidants, so that we can explore the effects of antioxidative properties or capacities on the processes of interest in the Tiger study. Additionally, we will also be able to look at other related metabolites such as glutamine, NAA, and choline. And just to note, for all of these spectroscopy studies that I've reviewed, as well as the data that I will be showing here today, glutamate, as well as the other metabolites we're looking at, are going to be presented as a ratio relative to total creatine. And that is because in the field, creatine tends to represent an energy marker that's relatively constant and is used as an internal standard for ratio calculation so that we can compare among subjects and across scanners. So first, we tested whether or not higher levels of peripheral inflammation, so specifically interleukin-6 and tumor necrosis factor alpha, were associated with higher levels of glutamate. And indeed, we found that higher levels of interleukin-6 were associated with higher levels of glutamate in the dorsal anterior cingulate cortex. And the statistics here show or represent this effect even after co-varying for relevant compounds, including age, sex, medication status, and the percentage of gray matter volume in the spectroscopy voxel that we prescribed. This effect size was smaller in tumor necrosis factor alpha, and unfortunately was not statistically significant. So we are finding some evidence for this pathway here. Next, we wanted to see whether or not inflammation and glutamate in the dorsal anterior cingulate cortex was related to connectivity, specifically in frontal cingulate limbic circuits. And while we did not find evidence of glutamate being associated with frontal cingulate limbic connectivity, we did find evidence of inflammation being related to structural and functional connectivity within these circuits. So what I'm showing you here is that white matter organization as indexed by fractional anisotropy using a diffusion MRI scan and applying deterministic tractography to trace the corpus callosum and the genu of the corpus callosum. We're finding that higher levels of interleukin-6 as well as higher levels of tumor necrosis factor alpha are associated with lower white matter organization in this white matter tract. And the data here are, the scatter plots here are the raw data, but the statistics depicted here are showing the effects even after co-varying for age, gender, medication, and tract length. Additionally, we're also finding evidence that higher levels of tumor necrosis factor alpha was associated with lower resting state functional connectivity in the frontal cingulate limbic network. And this was defined using a data-driven approach similar to my previous work in an independent cohort of depressed teens. So we are finding evidence of this link of higher levels of inflammation impacting the structural and functional connections of frontal cingulate limbic networks. But recall that another aim of the study was also to identify whether or not, or to test whether or not peripheral inflammation in glutamate may be contributing to depression recurrence. And we are acquiring data on self-reported levels of depression severity every other month in this study. And thus far, we've had 45 depressed participants provide at least 4 time points of data, which allowed us to apply a functional clustering algorithm to identify groups of participants on the basis of their trajectories of depression. And when we applied this method, we found that the majority of participants were grouped under cluster 0, shown here in black. And these are depressed teens who showed a relative improvement in symptoms over the 18-month period. A subset of the teens were grouped into cluster 1, which showed a worsening of symptoms over time. When I compared the two groups on baseline levels of inflammation and glutamate, I found that cluster 1, the group with a worsening of depression symptoms, showed significantly higher levels of glutamate in the dorsal anterior cingulate cortex and significantly higher levels of interleukin 6, as well as tumor necrosis factor alpha. I didn't show it here, but the effect sizes are actually the same. So while preliminary and the study is still ongoing, we are finding some evidence that inflammation in glutamate may be contributing to the maintenance and possibly recurrence of depression in adolescence. Next, we wanted to test if antioxidants may be moderating any of these associations. And as I mentioned earlier, our spectroscopy scan was unique in that we were able to estimate levels of glutathione as well as vitamin C. And we found that there was indeed a significant interaction effect between vitamin C levels in the dorsal anterior cingulate cortex and interleukin 6, and its effects on glutamate levels in the dorsal anterior cingulate cortex. So here, in individuals with higher levels of vitamin C in the dorsal ACC, we're finding an attenuation. So this higher levels here are shown in darker blue. We're finding an attenuation of the effect of interleukin 6 or inflammation on glutamate. However, in individuals with higher levels of individuals with higher levels of vitamin C, you're finding an attenuation of the impact of interleukin 6 on glutamate. In individuals with lower levels of vitamin C, depicted here in the light blue line, the dotted light blue line, you're finding that expected positive association. So higher levels of inflammation are associated with higher levels of glutamate in individuals with low levels of vitamin C. I actually find this plot easier to digest. So this Johnson-Newman plot allows us to look at the full range of a continuous moderator and its associations with the slope of our slope of interest. So on the y-axis here is the linear association between inflammation and glutamate, so interleukin 6 and glutamate. And on the y-axis are the range of values for vitamin C. So this is the exact same data, just plotted in a different way that I find more easily digestible. So essentially, for lower levels of vitamin C, you're finding a higher magnitude or a positive magnitude or association between interleukin 6 and glutamate. And for lower levels, you're finding an attenuated effect, meaning that the impact of inflammation on glutamate is not as strong or is not even present. So just to summarize the data thus far, consistent with brain models of stress in rodents, depressed adolescents are showing higher levels of peripheral inflammation, and that's related to higher levels of glutamate in the dorsal anterior cingulate cortex, which is a key hub of frontal cingulate limbic circuitry. And that, based on previous studies, we know that is a robust neurophenotype of adolescent depression. We're also finding that higher levels of inflammation are also associated with lower white matter organization and intrinsic functional connectivity in frontal cingulate limbic circuits. And of course, all of these data need to be replicated in larger samples, but it may be the case that higher levels of inflammation and cortical glutamate may be potential predictors of poor clinical course in depressed adolescents, and may have implications for understanding treatment resistance in this population. And it may also be the case that higher levels of antioxidative capacity or levels in the brain may buffer against the effect of inflammation on excessive glutamate in the central nervous system, and that may represent a key target for treatment, or even as an adjunctive for ongoing treatments in depressed adolescents. All right, so in terms of future directions, I'm very much interested in understanding how stress affects adolescent neurodevelopment to increase the risk for the onset and recurrence of depression. And I'm interested in performing studies that will allow us to manipulate stress levels and more tightly link how stress is influencing inflammation and influencing neurodevelopment. That would be captured in a more fine-grained manner than what I'm doing currently with Tiger, and how the course or trajectory of neurodevelopment, when shaped by stress and inflammation, impacts or alters the risk of the onset or recurrence of depression in this age group. Another area of research that I think is very exciting that I would love to discuss with folks here in the department is whether or not we can reduce stress and explore the roles of antioxidants or other mechanisms by which stress reduction may prevent the onset or recurrence of depression in high-risk teens. So these can include things such as direct anti-inflammatory strategies, obviously, antidepressants that may have anti-inflammatory effects. But this could also include lifestyle changes, particularly in teens who are not depressed but may be high risk for other reasons, exposure to early adversity or familial history. And it would be exciting to see whether or not things such as exercise, improved sleep, diet, or mind-body approaches are reducing inflammation and increasing antioxidative capacity and affecting these more downstream mechanisms or pathways on glutamate, and how that may potentially play a role in preventing or reducing the recurrence of depression in this age group. All right, before I end and answer any questions you may have, I want to thank and acknowledge the Tiger team at Stanford, and specifically the Stanford Neurodevelopment Affect and Psychopathology Lab, directed by Dr. Ian Gottlieb, without whom this work would not be possible. I also want to thank the UCSF Dolby Family Center for Mood Disorders, directed by Dr. Andrew Crystal. The Ray and Dagmar Dolby Family Fund has supported a portion of this work. And of course, I want to acknowledge funding sources, National Institute of Mental Health, internal grants at Stanford, and the Klingenstein Third Generation Foundation. This project really is the result of the literal blood, sweat, and tears of all of the members listed here. And so I cannot thank them enough. And we're excited to see more of the results from the continuous data collection. All right, so thank you all. And I do want to plug in this study. We are currently recruiting depressed teens for Tiger. There's a link here that takes interested folks to a screening form. If you have patients that you think would be good candidates, or if you know of any families that would be interested in participating in this study, please let me know, or please forward this link to them. And I look forward to answering any questions you may have. There's a link to my website as well as contact information here on the slide.
Thank you so much, Dr. Ho. This was a very comprehensive talk and very enlightening. Thank you very much for all the work that you've been doing. I'd like to open this up for questions. If you would, I think we have one question in the chat so far. If you'd like to unmute yourself, go right ahead.
Hi. I think Cher Rose just messaged me if you want to go ahead and unmute yourself and ask your question.
Okay. Hi, I was just wondering if when you're evaluating teens, if you're taking into account anywhere the use of recreational drugs, especially marijuana.
Yes, that's a great question. Are we collecting information and taking into account the use of recreational drugs in addition to anything else they may be taking, whether it's antidepressants, psychiatric medication, or even things that may be affecting our measures of cytokines? So that would include antihistamines, which a lot of them are on, NSAIDs. And the answer is yes. So we do collect that information. We are excluding teens who are engaging in substances to the point where they may be meeting criteria for substance dependence. But yes, there is a good contingent of our cohort that is engaging in marijuana or using marijuana and alcohol. And so that is taken into account in terms of statistical covariance.
Thanks, great talk, thank you.
Hi, Tiffany.
Hi.
This is Anne Glowinski, and I don't know if you know my name, but I've known yours for a very long time because I work with the Cingulate Third Generation Foundation as an advisory board member and a grant reviewer. So it is so exciting to hear about your program of research, and I can't tell you, how I think happy the foundation is also with having funded you when you were younger. It's fantastic. I have a question that is actually coming a little bit from the sort of what we know about chronic stress, right? And how it is also basically a predisposing factor to a number of terrible outcomes, including depression. And I was wondering if you have any idea, like, of sort of the relationship in us, like how do you conceptualize the relationship of acute stress in adolescence to basically chronic stress that a kid may have experienced their entire life? And I'm thinking, both about things that we think about all the time, like abuse or family conflict, but also things that we're just starting to think about, like being minoritized. Or having stressors that are more invisible to clinicians at this point?
That is an excellent question, Anne, and thank you for being part of the Klingenstein Third Generation Foundation. That pilot grant is what launched the entire Tiger study. So I am indebted to the foundation. The pilot data that I got from that grant is what I put into my KO1 application, and it was extremely well received by the committee. So I cannot thank you enough, and it makes me very, very happy to hear that, this is work that the foundation is supporting and is also excited about. So you had two parts to your question that I think are so important. One is the role of chronic versus acute stress and how that plays a role in sort of the conceptualization of these mechanisms and how they pertain to depression. And the other one is very related. I think the field has historically focused on dimensions of stress, including, the temporal nature of stress, chronic versus acute, as well as types of stress that are characterized by deprivation or neglect, the lack of positive input, versus threat and abuse, the presence of negative input. And I think, it's a lot I'm glad we have a lot of time for discussions. Let me answer the first question. With respect to chronic versus acute stressors, I think in the field, both in terms of animal models and human work, there's sort of this double hit hypothesis of stress and how it relates to mental health outcomes. So early adversity, which almost all of the participants in Tiger have experienced, is going to be affecting these brain circuits. And I can maybe move back, discuss this here. Primarily the frontal cingulate limbic circuits, and it's affecting the developmental trajectories of these circuits in a way that are going to impact how these teens respond to acute stressors later in life. And we know that adolescence is a time when there are a lot of unique changes and stresses occurring, particularly those characterized by social stress. And that, I think, leads to sort of a vicious cycle. I mean, I've talked about how stress is affecting the brain and how that then can impact behaviors that lead to depression or resemble depression. Depression itself is a stressor, so that is actually going to be feeding back and probably creating or generating more stress in that individual's life, which again alters the development of these neural trajectories. So I hope that answers that question. So early adversity is affecting the neurodevelopment of these circuits in ways that prime them to receive input during a period of heightened plasticity, where acute stressors may have more of an impact. And that would lead to this sort of vicious cycle in terms of maladaptive functioning and depression. In terms of types of stressors, so chronic and acute, but of course, deprivation, abuse, and being minoritized, these invisible stressors, which I think even some pieces of deprivation and neglect may fall under. These obviously matter, and these play a huge role, again, in, I don't want to say dictating, but influencing the environmental inputs that teens are receiving during a time when their brains are primed to be shaped by the environment. And the messages that they receive on a micro as well as macro level is going to be, incredibly important. And I think I don't want to be all doom and gloom, right? So that is the reason why we need to make systemic changes. That is the reason why we need to address disparities. We need to address the fact that our society currently needs to do a better job in terms of addressing all of these aspects of the environment, so that we have a chance for our youth to develop in the best way possible. So I hope that answers your question.
It was a tough question, and you did great. Thank you so much, Tiffany. And I mean, I think you summarized sort of the big themes. And I think obviously there are many important areas of research in these big themes to see both how we can do better and also how we can prevent, which may need to also happen, essentially way before adolescence, but then continue also, as you point out, during this critical period of risk.
Yes, absolutely.
Please feel free to unmute yourself. I'd like to ask a question.
Hi, Tiffany. This is Aoife O'Donovan, and I really enjoyed your talk. I'm sure you would guess that I would. And I just wondered about the specific considerations for translating this work in adolescence. So I know you've thought about it, but I'd love to hear what you're thinking in terms of targeting inflammation at this age. What would the risks and benefits be? And would you see that as being, primarily something we would do with preventive interventions or also with anti-inflammatory interventions? What do you think about that?
Yeah, that is a great question, Aoife. Thank you so much for attending this talk and being someone that I can, brainstorm with about the impact of stress and inflammation, more broadly on brain development and psychosocial functioning. So I think your question on what can we do in terms of taking this information and translating it into treatments or preventions? I'm not a clinical psychologist by training. I'm a cognitive neuroscientist. So I feel like I am lacking in terms of understanding what can be done on the treatment side. But I don't think it can hurt in any way to build healthy habits, to teach teenagers how to reduce stress, to engage in more exercise, to prioritize sleep. So this is not something we talked about at all in this presentation, but there are tremendous circadian changes that are occurring during this period as well. And teens, especially in this country, are chronically sleep deprived. And once you accumulate sleep debt, you really can't even get it back. And a lack of sleep is inflammatory. And that's something I would love to explore more. But obviously there are many different ways to attack heightened inflammation. And my goal is that we can try those different strategies, perhaps as adjunctives to current treatments, perhaps as preventative strategies, and seeing whether or not those things then lead to a normalization of downstream glutamate and a normalization or a pivoting of neurodevelopmental trajectories in a way that enhance brain health and psychosocial functioning. I would love to launch such a study, and it would be great to move forward in that direction.
Yeah, great answer. And I think maybe sometimes when we think about these molecular mechanisms, we jump to pharmacology, but actually there are lots of ways to build healthier immune profiles without going to pharmacological interventions.
Right, and I mean, one could also compare and contrast, but I it's maybe cheaper in some ways to at least bring in some of those lifestyle interventions. And those are things that you would probably want to do, or you would want your child to do anyway.
Great, thanks, Tiffany.
Tiffany, hi, this is Barbara. Could you speak a little bit about how does the intergenerational transmission of depression or the genetic load from caregiver to child impact how we think about mitigating stress from this neurodevelopmental framework?
That is an excellent question, Barbara, and very difficult to answer. But I will try my best. So one thing that is perhaps missing from my talk right now is the role of genetics, and of course the role of parents, being one major, I don't want to say gatekeeper of environmental experiences for youth, but certainly that is the case earlier in life. By the time a teen is an emerging adult, they'll have access to many different kinds of environmental exposure, peers, school, social media, for better or worse. But at least earlier in life, parents and caregivers really represent the primary source of input for a developing individual. And in terms of genetics, that's not something I've looked at personally. I think it is important to look at genetic factors that are relevant for inflammatory responses, and obviously the development of gray matter and white matter more generally, if at the very least to use them as covariates or to control for those factors so that you can see the effect of the environment and stress, and to get a better estimate of how stressors may be impacting the brain. It could be that I'm underestimating that effect, right, in my studies, or I could be overestimating, but regardless, it is a source of variance that I'm not currently controlling for. I do collect information on family history of depression, but the intergenerational transmission of depression and of stress and of trauma is not necessarily genetically driven. We know that teens who have caregivers who have been depressed during their lifetime are at higher risk for developing depression. But is that really due to a genetic load, or is it due to the fact that when you are raised by someone who is depressed, that is extremely stressful. That is very difficult. And in addition to experiencing probably greater negative input during that time, one may be lacking the positive input or support that teens are expecting, or their brain systems are really expecting in order to develop adaptively, right? So I think it's a very, very complex issue. It's something I would love to work on more. I think animal models here may be helpful too, in terms of getting at the translational components of these models and understanding exactly which pathways or which genetic factors or epigenetic factors may matter most. Yeah, it's a really great question, and something I look forward to diving into at some point.
Thank you for taking a stab at it. I really appreciate it, Tiffany. We have a question from Krista. If you're still here, you can unmute yourself and ask. Okay, I'll go ahead and ask then. Thank you for your great presentation. Do you expect or have seen that the changes in inflammatory responses relate to specific depressive symptoms or psychosomatic and or cognitive?
That is a very, very good question. And I think the field in general has moved towards understanding whether or not inflammation or components of inflammation may be underlying specific facets of depression, as opposed to explaining all depression symptomatology. And I have looked at that in my data. I don't have a slide for that right now, but I can send it around or add it at some point. But I am finding that if we group teens on the basis of endorsing symptoms of anhedonia or psychomotor retardation, anhedonia, psychomotor retardation, and somatic symptoms, those are actually the ones that show higher levels of inflammatory profiles, so specifically interleukin-6 and tumor necrosis factor alpha. That's very consistent with work by Andy Miller and colleagues at Emory, showing that higher levels of inflammation seem to be characterizing a subtype of depression that tends to be more presenting with anhedonia-like symptoms and psychomotor retardation. Although they're finding that much of this inflammatory subtype of depression in their cohorts are related to dopaminergic dysfunction as opposed to glutamatergic dysfunction. So I think the extent to which that is relevant in teens, I wasn't finding glutamate differences in my anhedonic versus non-anhedonic subgroup, but I was finding differences in inflammation. So it could be driven by a related neurotransmitter system process. Yeah, it's a great area for one to look at in the future, and I'm hoping to dive into that more.
Thank you. And Krista just said she has some noise in the background, so I don't know if you saw the thank you. We have a couple minutes left with Dr. Ho. Please feel free to unmute yourself if you have a question.
Hi, Dr. Ho. Thank you so much for your talk. I do a lot of work with schools, of course, and I really wonder about the message you would give to teachers, administrators. I can see, yes, kids need more sleep. These are policy decisions about what time in the morning they start, but there's a lot of stress through the pandemic and after as kids try to catch up with learning loss. I'm just thinking about, what kind of messaging can we give based on the science of inflammation and depression? Because I'm hearing teachers saying that anxiety and depression are huge right now.
Yeah, I think that is such a great question, Michelle. Thank you. Before the pandemic, academic stress was already such a big problem, as well as, the interpersonal stressors that teens tend to experience during this time period. And now, we're still in the middle of the pandemic, really. All of that is compounded, and it's gotten worse. So I think it's tough. What can administrators do? What can teachers do? I think I think it ultimately has to come at a policy or systemic level. We can try at the individual level to be more sympathetic, right? And to, promote practices that lead to healthy and supportive interpersonal interactions and prioritizing that. But I think now, unless something happens at a systemic level, it is really hard to see widespread change. And I can't imagine teachers already not doing that, or parents already not trying their best. But if the system, if society is not investing in the right environments in which kids are learning, and school is one of them, right? Teens and young children spend how many hours a day at school? If they're not receiving the right resources, right, from their counties, from the state, then how can we expect things to get better? So I'm sure that's not like the best answer here, but
Tiffany,
this is an urgent problem. We
Tiffany?
really. Oh yes, hi Marty.
I think for a person who's identified herself as a non-clinician, you've been doing clinically extremely well. And I would just add to what you were saying, the importance of identity and self-image as damaged by insecure attachments and early childhood adversity, and that helping people develop a sense of belonging and a sense of reflectance of their positive identity might be a help to the person who asked that question.
You are brilliant, Marty. Thank you.
That is an excellent note to end on today.
Yes, I love it.
That was a Michelle and Marty collab there. And Tiffany, you just hit the home run. So thank you so much for everyone and everyone's participation. It's been such a pleasure doing this series. It's wonderful to have Dr. Ho with us, and we look forward to inviting you back to hear more as your research grows and grows. So thank you so much, and have a great afternoon, everyone. Take good care.
Thank you. Bye.